This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 206, recorded on September 19th, 2019. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello, everybody.
Is it winter there yet?
You're kidding. We're doing okay.
This morning it was almost freezing here when I
really
at 6 a.m. Yeah, had to let the dogs out, and it was freezing, very cold. It's too bad.
We have 75 degrees for a change.
You know. As soon as August is over, it gets chilly. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
Where it's probably snowing already, right?
No, it's beautiful. We've got weather in the mid to high 70s each day, and then it cools off to, low 60s, high 50s each night. It's perfect.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Where it's 90 degrees, right?
No, today we have beautiful weather. We're having Michigan weather, but not as cool at night. But we're having Michigan weather today. There's a cold front that has moved through that's keeping our current hurricane Jerry, out to sea. So we're watching Hurricane Jerry. Hurricane Umberto is currently inundating the Bahamas. So yesterday we had not the Bahamas, the Bermuda. And yesterday we had a cruise ship come to town because it got diverted from Bermuda. So the city fathers were very happy. More tax revenue.
What a great name for a hurricane, Umberto.
Yes.
Remember?
And then we get Jerry.
Umberto Eco. Anybody read his books?
That's right. Oh, sure.
Wonderful.
Yeah.
Very good. If you like the work that we do here on TWiM and all our other podcasts, consider supporting us financially. Go to microbe.tv/contribute. We'll have more on that later. And also later, I'm giving away a book today, so stay tuned. From ASM Press.
Oh, great.
Today's snippet is brought to us by Michael.
Yes. I'm going to start today's snippet with a quote from Stan Falco, who unfortunately left us in 2018 because it's a sentiment that I think will set the tone. The good old days are now. We should not dwell on the past, to be sure, but we should not overlook the preface to it all because it still serves to teach us. It's the basis of current wisdom. So today's snippet is a remembrance of some of the early work of Stuart Levy, a pioneer in the field of antibiotic resistance in bacteria, and likely one of the earliest founders of the antibiotic stewardship movement. He died on September 4th, 2019, the day before we recorded TWiM 205. Vincent and I had occasion to interact with Dr. Levy on one of our earliest TWiM meeting shows in 2011 in Chicago at the Inner Science Conference on Antimicrobial Agents and Chemotherapy, known by its acronym as ICAC. And that was on the 22nd of September, 2011. For those of you listening to our back catalog, that would be TWiM 16.
Wow, ages ago.
Yeah. I still remember that we had this fascinating discussion with Dr. Levy along with David Livermore.
Right.
And Livermore is famous for his work on metallo-beta-lactamases. And he's professor of medical micro at Norwich Medical School and Public Health England. And our good friend Arturo Casadevall of Johns Hopkins University. And we all talked about antimicrobial resistance. And then finally, Arturo chimed in about why fungi don't cause disease. So it's really something to go back and watch us all interact. And it's a video as well as sound. But first, some of the particulars about Stuart Levy. He has a twin brother, Jay, who works on HIV. He's also a microbial scientist. And Jay's at UCSF. He's been working on HIV for a very long time. As well, he's got a sister, Ellen Koenig, who I believe is currently working at the Dominican Institute of Virological Studies in Santo Domingo, Dominican Republic, where her research interest is based on depression in response to antiretroviral therapy. So they have the three siblings are all in the microbial sciences. And Stuart left behind his dear wife, Cecile, and his 3 children, Suzanne, Arthur, and Walter. He graduated magna cum laude from Williams College and received medical degrees from the University of Pennsylvania. He completed his residency at Mount Sinai in New York, and he performed predoctoral research in Milan and then at the Institut Pasteur, which is going to be the basis of our discussion of his papers. He also did postdoc research outright at the NIH here in the United States. In an interview in 2015 for the Scientist magazine, when he was asked about his most influential mentor, he answered Tsutomu Watanabe. Quoting now Stuart through the article, "I think the biggest training I received was with Watanabe, and that was just for a summer. He was patient, methodical, and a master." And of course, Watanabe is probably one of the most famous individuals describing our plasmids or resistance plasmids, and I'll have something to say about that in a few minutes. And in the same,
I want to know if Elio hired him.
No, he hired me.
Very good.
I was his boss, but let me tell you, that's an original oxymoron.
Yes.
Because it's a contradiction in terms. Nobody can be a boss to Stuart. He was a very, very free-floating, free-flying individual, and the only good thing I ever did was to let him be.
Yeah. In that same Scientist article, when they asked him what his dream experiment would be. So those of you out there listening, start thinking of your dream experiment, especially if money were no object. And his dream experiment would be to design an experiment that would definitively and quantitatively demonstrate the link between subtherapeutic use in animals of antibiotics and the emergence of antibiotic-resistant infections in people. And he's probably responding to one of his perhaps most famous sets of experiments. This is the famous chicken feed experiment, which appeared in both Nature and the New England Journal of Medicine in 1976, where he showed changes in intestinal flora of farm personnel after the introduction of a tetracycline supplemented feed on farms. And this is probably the only prospective study out there to determine whether feeding farm animals antibiotics in their feed resulted in changes in the intestinal flora of farm dwellers and their neighbors. And he did this
Humans.
He did this farm dweller. I'm 2 hours early, so that's my excuse. He did this experiment with George Fitzgerald and Ann McCone, and basically he fed chickens a feed supplemented with tetracycline at a concentration of 110 parts per million. And as expected,
which is not much.
No, it's not much. 110 ppm is nothing. And within 1 week, the chicken's intestinal flora, and this was expected, was almost entirely TET resistant. And he saw increased numbers of resistant intestinal bacteria also appeared, albeit a little bit more slowly, in the farmers, but not in their neighbors close by to him. This was headline news in 1976 before CNN even existed.
And Michael,
yes,
sorry,
please.
if I could interject, you pointed out this was the first prospective study, but for our broad audience, many other retrospective studies have absolutely confirmed his observations and concerns.
which is also driving the removal of antibiotics from feed in many of the proteins that you can purchase at restaurants like McDonald's, Panera Bread. They're all going with the antibiotic-free raised chickens and all of these other sources of proteins that we consume. Later in Stuart, let me finish this thought about the chicken experiment. And within 5 to 6 months, around 30% of the weekly fecal samples from the farmers contained greater than 80% tetracycline-resistant bacteria, as compared to their neighbors that only had 6.8% of their fecal samples resistant to antimicrobials. And the neighbors, of course, didn't handle the feed or the chickens or their waste. The bottom line that they ended their New England Journal paper with was that it was selective pressure by the tetracycline-fed antibiotic-resistant bacteria that were in the chickens that were then somehow extending themselves to human beings that were in contact with the chicken, their feed. And this was just incredible in 1976. It was the beginning of the molecular biology era, so there was no high-throughput sequencing. Sanger was just beginning to work out Sanger dideoxy sequencing at the MRC in England. So it was a very different time. Later in his career, Dr. Levy responded to a challenge from Senator Ted Kennedy, where the senior senator from Massachusetts threw down the gauntlet to Stuart. And you have to have interactive with Stuart. When you challenged him, he would respond. And so Ted Kennedy said to him, if you're not going to save money, you won't get much interest in antibiotic resistance. And Dr. Levy took up the good senator's challenge, and this was a study that appeared in CID in 2009 that took a patient-by-patient analysis of resistance in a single Chicago hospital. And Stuart did this study with Robert Weinstein from Cook County Hospital. And their analysis showed a cost of about $21,000 per antimicrobial resistant infection per patient, producing a cost to the hospital of about $4 million and a total societal cost of as high as $15 million, including the loss of productivity. I didn't take the time to dig into the paper to see if this was just for $15 million for one hospital or whether they extrapolated it to the total hospitals in the U.S. And there are about 6,000 or so hospitals in the U.S., which raises the cost to $90 billion, and that would have been real money to Ted Kennedy. Mm-hmm. So Stuart joined the faculty, as we already said, at Tufts in 1979, where either Elio worked for him or he worked for Elio. And his hits, we're going to discuss the fact that he discovered the first active efflux pump involved in tetracycline resistant and the Enterobacteriaceae. And
let me just say something. Maybe it's premature, but let me say it now. The amazing thing about Stuart was that he crossed the gap between very heavy, beautiful science, basic science, and very applied stuff. There was no gap in his mind. He just went seamlessly from one to the other, and very few people can do that. It probably takes being trained as an MD to have that kind of facility. He just did that.
It's the translational research before it was made
before it was called that
by the NIH.
Right.
So,
but for him, it was, as I say, it was really seamless.
Yes, and I think it's this is why I picked the story I'm going to tell you today. I want to call to your attention some of his early contributions to our mechanistic understanding of how bacteria become resistant to these wondrous chemicals we refer to as antibiotics. It's a fascinating story that starts appropriately in Paris at an institute named for the individual responsible for the quote in 1854 in a lecture he gave at the University of Lille, where chance favors only the prepared mind. And that quote, of course, is from Pasteur. So here's the story. Stuart goes on leave from medical school, and he goes as a visiting research fellow, first to Milan and then to the Institut Pasteur. There he meets the giant Watanabe that we've already introduced you to. And I'm going to put into the show notes a Scientific American article that's a PDF from 1967 that describes some of Watanabe's contributions. And Watanabe is acknowledged as the father of our concept of resistance episomes, or plasmids as they were further than renamed, where I can imagine the encounter in a Paris cafe when something like this between Dr. Watanabe and the soon-to-be Dr. Levy. And so here's what Watanabe would have probably been telling him. Clinicians in Japan are increasingly alarmed to discover that Shigella flexneri, isolated from patients ill with dysentery, are multiply resistant to antibiotics. And Watanabe would have known that prior to the late 50s, isolates of the Bacillus dysentery bug, Shigella, were uniformly susceptible to most antibiotics available for treatment. Suddenly, these same bacteria were now simultaneously resistant to streptomycin, tetracycline, sulfonamides, and chloramphenicol. And then, to top, if that wasn't bad enough, some of the E. coli isolated from these patients' stools were also multiply resistant to drugs. The Japanese investigators working on this subject at the time also noted that some patients at the beginning of the treatment only had antibiotic sensitivity, but following the treatment, they excreted multiply resistant both Shigella and E. coli. So they knew something was unusual was happening in the intestinal tracts of these poor patients. And in the early years of antimicrobials, we didn't have that many drugs to employ against an infection. So it was like we're facing now. There's not a lot we can do when the bugs in your system that are responsible for the infection are becoming resistant. And Watanabe made all of this crystal clear in a review that was in Bacteriological Reviews in 1963 that then serves as the basis for the Scientific American article. But back to Stuart. The first paper I'm going to call your attention to is one where his lab went to show that an R plasmid, these resistance plasmids, which are very large plasmids on the order of an F factor. They're not like the plasmids that we use for routine cloning today. And the title of the paper was Detection of an Inducible Membrane Protein Associated with the R factor Mediated Tetracycline Resistance. And this appeared in Bbrc in 1974 and showed that new synthesis of an R factor specific protein occurs in E. coli mini cells. And Stuart was a master at working with mini cells. And mini cells are nothing more than a mutant E. coli that blebs off some of its cytoplasm without any DNA or other nucleic acid in it. And then you put a piece of nucleic acid into it, and you can ask questions. These were before cloning was developed. Mini cells were our cloning vehicles back in the bad old days of the 70s before cloning became famous. And so what was expected at this point in time was to appreciate we had figured out by now that a lot of antibiotic resistance was due to degradative enzymes like beta-lactamases. Yet at this point there were no degradative activities associated with tetracycline resistance. What was expected to be responsible for TET resistance, and this was the working hypothesis, that resistance might be explained by a decrease in the energy-dependent uptake of the antibiotic in the resistant microbes compared to the same cell without the resistance trait. They also knew at the time that resistance could be increased when the organism was pre-incubated in a small amount of drug, if you will, a sublethal concentration. And again, as the Levy lab were pros at using these mini cells, and there's a Nature paper that describes this in great detail that I'll also put in the show notes for you. So this bag of enzymes without DNA, they sneak a piece of these, they sneak the R plasmid into these mini cells and ask for protein expression, plus and minus pre-exposure to TET, and plus and minus the absence of the plasmid. So it's an elegant and fascinating paper to read, given our understanding and bias of the last 50 years of molecular biology. So the principal data elements in the Bbrc paper is one, a new protein band of 50,000 molecular weight is detected by SDS page, and it's only synthesized in the presence of TET, and that this protein was localized to the membrane. We now appreciate that this observation and the many papers to follow led Dr. Levy to discover that it was an efflux pump that was responsible for tetracycline resistance. It wasn't that the drug wasn't being pumped in. It was the fact that the drug was being pumped out. And that paper that he demonstrated that equivocally is his 1978 Nature paper, where he and his lab determined that plasmid-derived resistance to tetracycline involved in novel transport systems for the tetracyclines. Now, this family of observations about tetracycline resistance and efflux then led Dr. Levy and Dr. Walter Gilbert of Maxim Gilbert sequencing fame to found a company called Paratech Pharmaceuticals about 20 years ago, where Stuart's insight and fundamental observations into this efflux mechanism served them well in the development of one of the company's lead antibiotics, which is now a licensed drug in the United States, omadacycline, which belongs to the aminomethacillin subclass of the tetracycline antibiotics. But its hallmark is that it can't be pumped out, and it gets around many of the ribosomal mutations. Remember, tetracycline tackles the 30S component of protein synthesis, and it gets around those two mechanisms, two basic mechanisms of resistance to tetracycline. And omadacycline, I heard a talk recently on the wonder of this drug in the treatment of community-acquired pneumonias today. And that's one of the big issues facing clinical medicine dealing with infections. The other drug that Paratech has been developing is principally another tetracycline derivative, and that's against acne vulgaris. And acne vulgaris is a clinical syndrome that the uninitiated refer to as zits, but these are, of course, the very painful and very large comedones, papules, pustules, and nodules and cysts that result from the overgrowth of Cutibacterium acnes, obstruction and inflammation of your pilosebaceous units, or the hair follicles that accompany your sebaceous gland that we know as acne. And anyone who's got teenagers know that the dermatologist often gives you tetracycline creams or tetracycline pills in order to help your kids clear their skin infections. So, you know, Stuart was a very prolific on the prudent use of antibiotics. He testified many times before Congress on the subject of antibiotic resistance. And I think that he was really the impetus behind President Obama's starting the Presidential Commission on Antimicrobial Resistance back in 2015, which is a $1.2 billion initiative to combat antibiotic resistance.
I think this is a lovely homage to Stuart. I really enjoyed hearing you talk about this because he deserves it all. Well done.
Yeah, and I'd like to add to that. I was fortunate to be a postdoc in Elio's department at Tufts when Stuart was there, and he was a force of nature. I'm not sure I ever saw him standing still, but you always knew that he was working really hard on the front line, and he was just a great role model for the rest of us.
That's right.
Yeah.
It's beautiful.
I want to close with one thing. There was he'd sat for an interview for MedPage in 2014, and they asked him, what is the most memorable research published since you became a physician and why? Now, Stuart could have answered about his stuff, but instead he answered, the most memorable for me is the discovery that bacteria, Helicobacter, are the cause of peptic ulcers. This discovery has revolutionized our approach to a common disease, leading to a cure and no need for surgery. It is also a great tale for physicians about humility. We had told our patients for years that ulcers were because of stress or poor diet or type A personalities. Lo and behold, it's not the patient's fault. It's a bacteria. There's a lesson for all physicians here. And it really goes back to what Elio said earlier. He got translational research early, and he practiced it his entire career. It's a life well lived.
Thank you, Michael. I should have asked that of Elio when we interviewed him, right?
Yes.
We didn't ask him what's the most memorable scientific discovery in your career.
In your lifetime.
I think you would have said, there were too many to pick one. So many, right?
Sure.
I tell you, it was fun looking at this. I enjoyed reading all of the papers. I so much enjoyed going back and reliving mini cells. I worked with mini cells when I was a postdoc, and they absolutely fascinated me. I mean, the young people don't appreciate the what the kit technology has offered them today in getting expression of proteins and colors. A lot of this stuff was done with radioactivity, and it really is something.
All right. That's great. Thanks, Michael. All right. We have a paper now which was suggested by a listener. Simon wrote, I would love to hear your take on the paper. The title of it is Bacterial C-gas-like Enzymes Synthesize Diverse Nucleotide Signals. In Nature, it's a fresh paper that has an unimaginable amount of data. Each section feels like it probably could be a paper on its own.
That's what I was going to say.
The conclusions are dramatic and impactful, and the figures are all scientific Picassos. Love the show. Okay.
Now, is that a compliment that it's a Picasso?
Oh, absolutely. I think so. I love Picasso. It's great stuff. You don't have to love him, but the work is very cool. So you heard the title. This is from Whiteley, Eaglesham, De Oliveira Mann, Morehouse, Lowy, Niemingen, Daniel Chanka, King, Lee, John Mechelanos, and Philip Kranzush. And this comes from Harvard Medical School, University of California, Berkeley, Brandeis University, and that's it. So if you want to know a little more about Philip Kranzush, he was a guest on TWiM many years ago when he was a PhD student at Harvard. Oh. And now he's a faculty member. He left and came back. So this paper is all about cyclic nucleotides. And
it's an open access paper.
Very good. Open access in Nature.
Yes.
Second messengers. We call these molecules second messengers because cells can use them to amplify signals and control other responses. And I am most familiar with them because not too long ago in human cells, it was found that when DNA, double-stranded DNA, gets into the cytoplasm of a eukaryotic cell, it is sensed by an enzyme called cyclic GMP-AMP synthase, which we abbreviate as CGAS, and it synthesizes a cyclic dinucleotide, cyclic GMP-AMP. So think of a guanosine monophosphate and adenosine monophosphate. They're linked to form a small circle.
By the phosphate.
By the phosphates. And this then goes on to bind to a second protein in the cytosol, which ultimately leads to the production of interferon. The assumption being that DNA in the cytosol is foreign. There shouldn't be any DNA in a eukaryotic cytoplasm. And therefore, the cell reacts by producing interferons, which would then inhibit virus replication. So when that was discovered, not too many years ago, everyone said, aha, this also happens in bacteria, because cyclic dinucleotides hadn't been found in eukaryotic cells, but it has a long history in bacteria. In fact, these were first identified in bacteria. And that's why when they were found in mammalian cells, eukaryotic cells, we understood their importance in signaling pathways. And nearly every bacteria phylum encodes some kind of cyclic dinucleotide signaling pathways. And so far, these dinucleotides are all made with purines, all right? A's or G's. No pyrimides have been found so far. And these, what do? They control a whole range of responses, like transitions between planktonic and sessile growth, osmoregulation, cell wall homeostasis, DNA damage responses, chemotaxis, virulence. So you get the picture. These molecules can control a lot of things. They're made, and then they go on to control lots of other pathways. And in fact, it's just a little funny here that the pathway I just described you in mammalian cells also senses bacterial CDNs, cyclic dinucleotides.
It makes sense. It's chemically the same thing.
Yeah, so if it gets into the cell, then it's sensed and activates the same pathways. And so in this paper, what they do is say, let's discover as many as we can of these cyclic dinucleotides. And they start with Vibrio cholera, whose genome encodes a dinucleotide cyclic cyclase, we'll call it, and which is encoded by a gene. And it synthesizes cyclic GAMP, similar to the one we just talked about in mammalian cells. The enzyme is on a mobile genetic element that can be horizontally acquired. It's found in all pandemic isolates. And they were studying this, and they found unexpectedly in E. coli a operon where this gene from Vibrio DNCV is replaced with a gene whose function is unknown. And they said, well, this could be a CDN synthase. So they did an experiment. They purify the protein from E. coli, which is called CDNE, cyclic dinucleotide E. They incubate it with P32 labeled triphosphates, and they do a method that
Stuart Levy would have done.
All of us have.
Hey, I've done that method.
We've probably all done it.
We all done this method.
TLC, thin layer chromatography. Not many people do anymore. You look at the reaction products when you put each of the labeled bases in, and it made something that was different from all the known cyclic dinucleotide. And so they mixed different labeled triphosphates together. And it ends up being that the product of this E. coli CDNE gene is cyclic UMP-AMP. So it's a hybrid purine-pyrimidine cyclic dinucleotide, which they hadn't seen before, which is, of course, great reason for just encouraging people to look. Just discover things. Look. You look, seek, and ye shall find. And so this enzyme in Vibrio DNCV is a structural homologue of CGAS, which is the mammalian enzyme that makes the cyclic dinucleotides. But it doesn't have overall sequence homology, but it does have some conserved amino acids, 4 amino acids, which look like they might be active site residues, and they show that if you change them, they destroy catalysis in the enzyme. And they can show that the E. coli enzyme, which I just described to you, is probably ancestrally related to both cGAS, which is, again, the mammalian or eukaryotic enzyme, and the Vibrio enzyme. So they call the E. coli gene cDNE, cGAS DNCV-like nucleotidal transferase in E. coli. Now, in Vibrio, this cyclic dinucleotide synthase controls the activity of a phospholipase. And often these cyclic synthase genes are next to other genes that they regulate. And so in this E. coli gene, cDNE is next to a phospholipase. And they show that it's, in fact, that phospholipase can be activated by the product of the E. coli gene, which is the UMP-AMP that we just talked about.
It says in the paper that it encodes a patatin-like phospholipase.
Yes.
I never heard of patatin.
Patatin. Maybe, does Michael know?
No, I had to look that word up too.
A glycoprotein found in potatoes.
Yes.
Yeah.
So it's a patatin. Yeah, I guess it makes sense. Patatin.
Yes.
It's
about French fries.
It's all about French fries. That's right. Anyway, so that is somehow controlling that enzyme. Exactly what's going on there, we don't know. But so the idea is that specific dinucleotides.
Hey, patatin is found in potatoes.
Yes, it is.
That's why it's about French fries.
Maybe we have a title there, Michael. Yeah.
It's about, I think Michele said about snacks.
It makes up 40% of the soluble protein in potato tubers. Just so.
But we digress.
We digress.
Which we never do, right?
No. This is not TWiM.
Well, the point here is that these cyclic dinucleotides can control different enzymes. And in this case, it's this patatin-like phospholipase, but it can be other things as well. And in fact, you're going to see that they discover a lot of these enzymes, and what they're controlling we don't know, but that is for everybody to discover if they'd like to. So,
but I think they point out a number of the known effectors are kind of aggressive molecules, toxins, enzymes that digest perhaps host proteins, et cetera.
Right.
And it stems out of the Vibrio and the fact that they discovered it off of one of these pathogenicity elements.
Yeah.
Right.
That's right. They solve some structures of these proteins. In fact, they do that quite a bit in this paper. They do X-ray crystallography. They have to purify the protein and get it to crystallize and then solve its structure. And because these are relatively small proteins, so they're not well suited to cryo-EM. So they solve crystal structures, and they do that for a cyclic dinucleotide, a cDNE homolog from a thermophilic bacterium called Rhodothermus marinus. And they can see from this that the enzyme looks like a polymerase beta, a DNA polymerase. It has a nucleotidal transferase fold that's very similar. So there's some ancestor probably to both this DNA polymerase beta and these cyclic dinucleotide synthases. So they're really nucleotidal transferases. Of course, they don't have templates, though. And one of the interesting things here is that in some cases, these enzymes are active without having a ligand in them. So, for example, there are similar enzymes in eukaryotic cells that are activated by conformational changes caused by binding nucleic acids. But the ones we've talked about so far, cDNE in particular, is active without having a nucleic acid. It will make these cyclic molecules if you just add them to it. It doesn't have to have a nucleic acid in it. So they make structures of this thermophilic bacterium cyclic dinucleotide enzyme with non-hydrolyzable substrates. So remember, that takes ATP and UTP and makes a circle out of them, but they can use non-hydrolyzable triphosphates, so it won't make a circle. They will simply bind to the enzyme, and then you can see what side chains are important. And they identify an asparagine that makes a hydrogen bond with the uracil and helps position the ATP alpha phosphate for attack by the 3' hydroxyl. So this asparagine apparently is very important for this catalytic activity. And it's located in, they say this is probably a really important residue, and they think it would dictate product specificity of the enzyme. And it's nearly, when they look at all the known cyclic dinucleotide homologs, at least CDNE, they see that this asparagine is nearly universally conserved. An exception is an enzyme from Elizabethingia meningoseptica, nosocomial pathogen, which has a serine at
named
this
after the famous pediatrician scientist Elizabeth
right.
King,
who we've mentioned before, I'm sure. Mm-hmm. Right? Yeah. Has a serine at this position, and this enzyme from Elizabethingia makes cyclic dipurines instead, and they solve the structure of that enzyme as well. And if they put asparagine into that enzyme, it makes it make pyrimidine-containing products as well. So this position, which is asparagine in some, serine in this one particular bacterium, is really important for making the specific product. And all you have to do is change that one amino acid to reprogram it between purines and pyrimidines.
That's amazing.
It's very cool, isn't it?
Yes.
Sure is.
They do some more structural comparisons, which between the enzymes we've been talking about, CGAS, DNCV, 2-CDNE homologs, and also OAS1, which is another eukaryotic synthase. And they say have three features. One, they have a common DNA polymerase beta-like protein fold. Even though their sequences are very, very different, they all fold into an enzyme that looks like that. And second, they have template-independent synthesis of these small molecules. And third, they have a active site that allows making different products by simple amino acid substitutions. So they have a name for this family of enzymes. They're called CDNTases, CGAS DNCV-like nucleotidal transferases.
Oh, I wish the nomenclature were a little bit simpler, but
CDNTases. Yeah, well, what can you do?
They're probably closet immunologists. They just take the first two names, take their initials, and so cyclic dinucleotides, and then they stick the ACE on it.
Yeah, and nucleotidal transferases.
Yeah. Transferase, yeah. It's a logical name.
Makes sense.
Yeah.
Now, they're interested next in, many bacteria live next to or near eukaryotic hosts. So can these be detected? Can bacterial products be detected? And they look at STING, which is a eukaryotic protein that recognizes the product of CGAS. Remember, CGAS is activated by DNA. It makes cyclic GAMP, and that cyclic GAMP binds this protein called STING, and that induces interferon. So their funny cyclic molecule CUMP-AMP is recognized by STING. In fact, mouse STING binds multiple cyclic dipurine molecules, but STING didn't recognize CUMP-AMP, only the purine, cyclic dipurine molecules. So there is some specificity there. There is another mammalian cyclic dinucleotide sensor protein called Recon that was discovered after the CGAS story, and that can recognize this cyclic UMP-AMP, which is interesting. So they found a receptor for this purine pyrimidine in eukaryotic cells, and so maybe that is involved in recognition of bacteria, as we suggested earlier, and that's something that I'm sure they'll be doing as we move forward. So there's a common ancestor to these enzymes, most likely, and they say, well, we probably only know a few of them, so they do a kingdom-wide analysis to try and find more of them.
This is where the Picasso reference comes into play, because their figure 4, which you can see because it's in open access, is the Picasso.
That's the Picasso, this little phylogenetic tree of everything they discover. So they do a little bit of bioinformatics, and they do some biochemical screens, and they find over 5,600 unique bacterial enzymes that look like they are these CD-NTases. They have the right structural features. They were found in over 10% of bacterial genomes whose sequences are available. They span at nearly every bacterial phylum, including human commensals, pathogens, extremophiles, and agriculturally important bacteria like rhizobia and plant pathogens. Everywhere. I guess they're important, right?
I was amazed by that.
Now, if that's not enough for you, then they go ahead, they purify 66 of these proteins. That's not trivial.
No.
I don't think one person could do that. I think you need to have multiple, because you have to take the gene, the DNA, and you have to put it in an expression system, and most of the time it doesn't work, and you have to tinker with it, and then you have to purify it, and then you have to test it for nucleotide product synthesis. So you have to add labeled triphosphates and then do thin layer chromatography.
So that you just do the math, folks. That's 66 genes into 66 expression vectors into 66 radioactive labelings for the TLC plate. Them's a lot of experiments.
Mm-hmm.
Yeah. To say this paper is comprehensive does it a gross.
Well, the thing is, there aren't that many. There aren't that many authors. We've seen papers with more authors. So kudos to all of you. I'd be curious if any of you are listening, how many people did the protein production, if they divided it up among 3 or 4 or 5 people. I'm just curious. Anyway, most of these enzymes didn't have activity. No activity was observed from some, but they found 16 of the most active ones out of the 66 that they tried, and figured out what they made. And they have one that they highlight in particular from Legionella pneumophila.
Yay!
I thought you would like that.
I did.
It makes a cyclic dipyrimidine, like cyclic di-UMP. And this enzyme has an asparagine residue, which they say was in many of these, but not all of these enzymes. Then they also use mass spectrometry, coupled with using different labeled triphosphates to find the products. From these 16 highly active CD-NTases, they found enzymes that produce 7-purine pyrimidine and purine pyrimidine hybrid combinations. So they make a wide array of signals as well.
And I can interject here that Aaron Whiteley is the lead author on this paper. He's a postdoc on the team, and he worked with Karina de Oliveira Mann on the team and recalled one day where the two of them were looking at the mass spec results, and they didn't see any of the standard signaling molecules come off the results, but they saw these masses of oddball cyclic dinucleotides that they then realized after doing more analysis were cyclic trinucleotides. And it was just a mind-blowing experience.
Yeah, so one of the ones they get are trinucleotides, not dinucleotides. And this, so this came from some, as you said, where they couldn't identify the expected dinucleotides. So they looked at it a little further. And the one they find from an Enterobacter host, Enterobacter cloacae, is an AMP-GMP cyclic trinucleotide from that one. And they find
what's next? Tetranucleotides?
Well, if you think about
that'd be it, though. No more.
If you think about what's going on here, you have these dinucleotides that are effectively serving as a key inserted into a lock to result in expression. And all they're doing is literally rekeying a lock. And they're effectively saying, in order to turn this set of genes on, the specific circumstances have to be such that we need a different pin in our key. And so now you got the tricyclic nucleotide to make that key to turn on the gene. And it really is going to architecture. It's going to depend on the circumstance. And I think the exciting thing that we're going to see is what are the circumstances that cause these genes to turn on, and that will give us an idea of the microbial ecology of what's going on. And specifically, I'm curious as to what it's physiologically doing in the Legionella.
Yes, these are the questions.
That's well put.
They say that larger cyclic oligonucleotide products may be more common than we expected. So maybe they would get even bigger than a 3 or a 4. Who knows? You have to look for them. This particular one, though, this cyclic AAG, is not recognized by Sting, but it is recognized by Recon, which is the other mammalian nucleotide recognition protein that we mentioned. So already in mammalian cells, you have proteins that will recognize 2 but not 3. So it's very interesting. These genes encoding these, by the way, are on mobile genetic elements. And so they can move around from host to host and depending on who needs what. And they're usually adjacent to the effectors that are controlled, like we said, the phospholipases and the nucleases, pore-forming agents, as Michele said earlier. So sorting out what these are doing is going to be very interesting, I think. Well, I think that's about it here. So they're widespread. These CDNTases are widespread. They make a different, diverse range of dinucleotides, trinucleotides, and who knows what more. They're on mobile genetic elements, so they can move around, which is quite interesting. And they say understanding what these do is a major challenge. And we have to see what they're doing now. But who knows?
It seems they've opened a whole new field, haven't they?
They have opened a field. So I'm sure they're going to be involved in pathogenesis. And so they close by saying the possibility that diverse CDNs and related nucleotide signals produced by prokaryotic CDNTases act as agonists and inhibitors of innate immunity and other host metabolic pathways provides an important new reservoir of compounds with biotechnological and therapeutic applications. Anyway, it's like a
that's a profound statement. That's a profound statement.
It is. It's a whole new area opened here, and many people can get these and start working on them in their favorite organisms and see what they do and devise ways to figure it out. So it's quite seminal, I would say. Yeah.
Well, it goes to the last line in the first paragraph of their paper. Recently, synthetic cyclic dinucleotide analogs have emerged as promising lead compounds for immune modulation in cancer immunotherapy. Yep. So the bacteria have already done the R&D for billions of years, showing us how to make these things and engineer them properly to get the effector, especially if you're trying to inhibit innate immunity or activate innate immunity. So it could be something really significant.
And actually, the authors have been wise to that. They have filed patents on the nucleotidal transferases and also the structure of the complex and the uses thereof.
Have they made a company?
I can't tell. I don't know that. That's a good question. But let me tell you a little more about Aaron Whiteley. So he is a Jane Coffin Childs funded postdoctoral fellow and mentored by both John Mekalanos and Philip Kranchuk. So he got a bachelor's degree in biochemistry and molecular biology at UC Davis, and he did some research there as well. And from there, he went and was a fermentation engineering technician for a bit, and then was a staff research associate at UCSF with Leanne Baxter-Lau, where he did some work on bone marrow transplant diagnostics, etc. He also was CEO and founder of a company called Bench Connect, which was a startup software company to develop digital lab management tools. He was motivated to go on in his career. So he was a graduate student at UC Berkeley, and he had every intention of studying immunology. But he found himself in a seminar class taught by Dan Portnoy, who talked about bacterial pathogenesis, and Aaron says he was an instant convert. Dan has a gift for making small experiments tie into larger concepts in biology, and he was also a great unifier who built terrific teams both in his lab and in the greater Bay Area, and he had terrific trainees. Aaron was especially grateful for what he learned from Josh Woodward, Michele Rainier, and J.D. Sauer, who are all now leading their own research groups. He also learned from Dan that you want to think about larger questions like evolution. You want to explore general concepts, make sure the questions you're asking are broadly relevant, and also be rigorous. Design experiments to prove yourself wrong. So then he decided to go to John Mechalanos' lab for his postdoctoral training because he was just so impressed by the many genetic screens that the lab has done over the years, and credits John's creativity with that. And he was delighted that he was able to do his postdoc and have these two mentors and Philip in particular, Kranchos, taught him, introduced him to the world of biochemistry and how to work in a cross-disciplinary way. And we clearly saw that in this paper. He said all of the authors on the paper made really important contributions, and it's been just a great experience. He also has some advice for junior colleagues. He says, don't worry about making the right decision. Go with your gut, take risks, be open to what you don't know, and you'll see where the science takes you. He also is pleased that he and his wife, who is also a scientist, were jointly recruited to the University of Colorado. They'll be starting their faculty positions there in January, and they'll be bringing with them their newborn son, who as of today is 3 days old.
Wow. Cool.
Along the way.
So he responded to you within days of his child being born.
Within 1 day of his child born. And along the way, he has earned a number of awards, Outstanding PhD Leadership Award. He also was recognized as the Outstanding Postdoctoral Fellow at Harvard Medical School in his department, and has also had a number of other leadership positions, including he was co-chair of a Gordon Research Seminar on microbial toxins and pathogenicity, and was president of a student graduate group. So he's had just an amazing career so far, and keep your eye out for Aaron Whiteley. He is going to continue to make great contributions to our field, I am certain.
Nice. Thank you.
Right.
Thank you, Simon, for suggesting the paper. And TWiV 148. That's the TWiV where Phil Kranzush was our guest. That was recorded on September 11th, 2011. And I love it when you see PhD students who then go on and have their own labs and do really cool stuff, right? It's a great validation of things working the way they should. All right, we have two interesting emails. Let's see if we can answer them. They're both from Ben. Ben is a PhD student at the University of Adelaide. Of course, that's in Australia. And the first one, Dear TWiM holobionts. I wanted to see what you thought about the term reduced genome size as applied to organisms whose genomes are small due to reliance on other organisms for their survival, such as Rickettsia. The idea that having a small genome is because there's lots of metabolic processes, etc., that you rely on other organisms to perform for you has never sat well with me. Many plants have wildly large and complex genomes, but are utterly dependent on yeast and bacterial symbionts for their survival, while us humans could never have survived without our microbes before modern medicine. Should then we, the plants dependent on symbionts, and the countless number of similar examples be considered as having reduced genome sizes as they cannot survive without genes that are encoded by their microbes?
That's a Talmudic question for Eli's blog.
It sure is.
It's a good point. My take is, all right, as an outsider, right, the ones we call reduced are the ones where we could sort of see the history of reduction, right? And you could see very, very small genomes and what they once had, what they get from their organisms. So we can't really tell what we have lost over the years. Well, maybe we never lost anything, right? 'Cause we evolved in the presence of microbes, so we didn't lose anything.
We also are evolving at a much slower rate than your typical microbes, so maybe fewer chances for that trial and error of loss.
Yeah.
The selective pressure may be qualitatively different. I'm not sure I know what I'm saying when I'm saying that, but it does seem to me that it's maybe apples and oranges.
It's a provocative question, though.
It is. It's a good question.
I don't know. I just think from a human viewpoint, I think we evolved in the presence of microbes, so we only had what we needed to survive in their presence, whereas an endosymbiont, is free living and then goes into another host and then loses things over time. I think it's kind of a different situation. It's kind of related to what Michele said, I think. By the way, Ben says it's a chilly 15 Celsius in Adelaide, South Australia, but 32, only 200 kilometers northwest where I grew up. Hope you were all safe during Dorian. Keep up the wonderful work. Okay, so then Ben writes, sorry for the bombardment of questions, but I found this one too interesting not to ask. It is not a problem, Ben. We like questions. Mollusks and arthropods use hemocyanin rather than hemoglobin to transport their oxygen. Hemocyanin uses copper to bind rather than iron and is free-floating in the hemolymph rather than packed into red blood cells. It's thought that the evolutionary reason for this is that in cold waters with low oxygen, copper is more efficient than iron at binding oxygen. But this doesn't explain why arthropods that live in warm climates like spiders use hemocyanin. So my question for the TWiM team is, could hemocyanin have been selected for in warm, oxygen-rich environments due to the antimicrobial effects of ionic copper? Looking forward to seeing what you all think. What do you think, Michael?
That's a question from Michael.
I don't know the concentration of ionic copper associated with the hemolymph. Metallic copper is effectively the large leveler for its antimicrobial activity, and it has to do with it stealing electrons from the microbe and moving them through the metal. Yeah. The ionic copper resistance mechanism, bacteria have evolved mechanisms and pumps to rid themselves of it. So my guess is why hemocyanin or hemolymph was the hemolymph hemocyanin was selected is probably part of the electrochemical properties of copper and its ability to hold the oxygen in that scaffolding effect, the coordination chemistry of the copper. I have to go and look at the structure of the hemocyanin to try to think about it, but it's a good question.
Yes, Ben, so thank you. And if anyone else wants to weigh in, please do on both of Ben's questions. And if you have a question, please send it in, TWiM at microbe.tv. And that's it for TWiM 206, where you can find our show notes at microbe.tv/TWiM or at asm.org/TWiM, either place. And if you listen on a phone or a tablet on a podcast app, please subscribe to the show so you get every episode. And in turn, we know how many people are listening. That's important to us. If you really like what we do, go over to microbe.tv/contribute. We have ways using PayPal or Patreon where you could give us a certain amount of money a month, as little as $1 a month. If most of you listeners did that, that would really help us a lot with our expenses. And as I said, send us your questions and comments to TWiM at microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter is at Small Things Considered. Thanks, Elio.
My pleasure.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. I'd like to give away a copy of the ASM Press book called The Bacterial Spore, From Molecules to Systems, edited by Adam Drix and Patrick Eichenberger. If you would like this book, lovely book, everything you ever wanted to know about spores, bacterial spores, please send an email to twim@microbe.tv with the subject line spore. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.